Right. Let's unpack this.
Hmm.
Today, we are focusing on one of the most critical and I think frequently misunderstood relationships in all of behavioral neuroscience.
I would agree with that.
And that's the fundamental link between chronic, just unrelenting stress and the status of our prefrontal cortex, the PFC.
And that is the absolute core of executive function. We often call the PFC the brain CEO for a good reason.
Right, the command center.
It is the command center.
Yeah.
And if the CEO goes down, if that top-down regulatory power is compromised, the whole operation, the entire company of the mind, it falters. And that is what we feel is burnout.
That is precisely the mission for this deep dive. Our listeners going deep into the foundational sources here, specifically the seminal work by Dr. Arnsten and her colleagues, but also these critical human neuroimaging follow-up studies.
Right.
We need to understand the precise neurobiological mechanisms, the structure, the signaling, and the circuits that link the stress state directly to cognitive collapse.
And we're not just talking about feeling tired. We are looking for verifiable physical and functional changes.
The hardware and the software?
We need to identify the hardware failure points and the software failure points in, excruciating detail. We'll use analogies, we'll use metaphors, but we have to stick rigorously to the correct neurobiological terminology.
Absolutely, to maintain accuracy. We're aiming for the kind of understanding where you can actually picture the molecules doing the damage. We know that chronic stress isn't just unpleasant. It's a known risk factor for major neuropsychiatric conditions. We're talking severe depression, anxiety disorders, addiction.
And it specifically disrupts those core PFC functions like flexible problem solving, emotional regulation, and of course, working memory.
The challenge here then is integrating all the data. We have these beautifully detailed mechanistic findings from animal models.
Right, often chronic restraint stress in rodents.
Exactly, and that reveals the structural damage at a cellular level. But then we need to connect those microscopic findings to what we see in human neuroimaging.
And that's where we leverage these powerful studies like Liston et al. From 2009, which explored how, and this is the title, psychosocial stress reversibly disrupts prefrontal processing and attentional control.
That cross-species translation is what makes this so powerful. So let's start at the very beginning, before the chronic damage even sets in.
Okay.
We have to understand the immediate or acute mechanism. How does the brain's executive control center modulate its performance just in the presence of stress hormones?
This is where the story of the PFC really begins. That CEO region doesn't operate in a vacuum.
Right.
It's constantly being modulated by these internal signals. And the most important of these are the catecholamines, dopamine or DA, and norepinephrine, NE.
These are the classic alertness and drive neurotransmitters, right? We associate them with fight or flight, but also with focus.
Exactly. And the seminal work here, especially Arnsten's 1998 paper, Catecholamine Modulation of Prefrontal Cortical Cognitive Function, established that these neurotransmitters act like an incredibly complex, high-resolution tuning knob on the PFC.
A tuning knob.
Yeah.
I like that.
They are absolutely necessary to enhance function, but, and this is a profound caveat, they must be at precisely optimal levels.
So if the PFC is a high-performance engine, DA and NE are like the fuel injection system, but the engine is super sensitive to the fuel mix.
That's a perfect analogy. Or think of it like tuning a delicate radio signal. You need that optimal frequency. If the catecholamine signal is too low, the PFC is sluggish. The network just struggles to maintain focus, and you lose signal clarity.
But the danger isn't just too little, is it? The danger in stress is that the tuning knob is suddenly cranked up way too high.
Precisely. In a high-stress scenario, when you have an excessive monoamine release, a flood of DA and NE, that signal is no longer enhancing performance.
It's just noise.
It's jamming the system entirely. And that's the core tension. DA and NE are both the PFC's best friend and its greatest vulnerability.
Okay, so let's talk about the specific hardware that mediates this optimal tuning. Which receptors are involved in keeping that signal strong and clean?
So the magic happens through very specific cellular architecture. The enhancement of PFC function, that sweet spot of focus and cognitive power, relies on the stimulation of two specific receptor types.
Okay.
The D1 dopamine receptors and the alpha-2 adrenergic norepinephrine receptors. When an optimal amount of catecholamines hits these specific receptors, the functional communication within the PFC networks strengthens.
This sounds incredibly intricate. We need to understand how the D1 and the alpha-2 receptors physically translate that chemical signal into strengthening the network. What's the key pathway they manipulate inside the neuron?
This is where we get into the, the real nitty-gritty of the neurobiology. It's called the CAMP-HCN channel signaling pathway.
Okay, let's break that down.
This pathway is the critical internal mechanism that determines whether a PFC neuron stays stable and active or if it becomes quiet and inactive.
Okay, so let's simplify those acronyms. Tell me about CAMP-MP first.
Right, so CAMP-MP, or cyclic AMP, is an internal cellular messenger. You can think of it as the neuron's internal maintenance crew.
A maintenance crew, okay.
Or a secondary signal that carries instructions from the surface of the cell deep inside. Now, when the cell is stressed or inactive in certain ways, this maintenance crew, CMP, gets really busy. High levels of CMP then go on to activate these protein structures on the neuron's membrane.
And those protein structures are the HCN channels.
Correct. HCN channels are protein gates, or ion channels. And when they get activated by high levels of CMP, they open up.
And what happens when those gates open?
They cause the neuron to become hyperpolarized.
Hyperpolarized. That sounds like the opposite of an action potential, the opposite of firing.
It is. Hyperpolarization means the neuron's internal electrical charge becomes significantly more negative.
Okay.
Think of the neuron as having this baseline electrical charge it needs to maintain to be ready to fire. When it hyperpolarizes, it drives that charge far below the baseline. And this effectively silences the neuron.
It just makes it incredibly difficult for it to fire.
Exactly. It can't generate the stable, focused action potentials that you need for complex thought.
So if the neuron is quieted, it can't maintain those persistent firing patterns that you need to hold, I don't know, your dinner plans or a complex problem in your working memory. The thought just collapses.
Precisely. That stable, persistent firing is the cellular foundation of working memory and top-down control. So the key mechanism for enhancing PFC function, as detailed in work like Wang et al. In 2007, is that the D1 and alpha-2 receptors, they inhibit that maintenance crew.
They suppress CAMP.
They suppress CAMP. They stop the internal messenger from issuing the instruction to shut down.
So they are effectively closing those inhibitory gates, the HCM channels.
That's it. Closing the gates prevents hyperpolarization, and that allows the PFC network to maintain that stable, persistent firing that is so essential for strong working memory. The optimal level of catecholamines acts as a highly effective filter, preventing internal noise from disrupting the signal.
That is just remarkably precise and elegant. They're turning off the neuron's own self-silencing mechanism, which makes the network resistant to interference and distraction.
But here is the immediate dangerous flip side. This delicate balance is immediately broken during what we call acute stress overload. And this is the phenomenon first explored by Arnsten and Goldman-Reichert way back in 1998.
So this is when the tuning knob is suddenly turned far past optimal.
Far past it. That excessive monoamine release, that flood of dopamine and norepinephrine during a sudden uncontrollable stressor has the complete opposite effect. It doesn't enhance, it jams the system entirely. It just overwhelms the circuit.
How does the mechanism switch from helpful to harmful so quickly? Does it start hitting different receptors?
It does. While the D1 and alpha-2 receptors are the key to that optimal zone, when catecholamine levels spike excessively, they begin to heavily engage receptors with lower affinity.
Okay.
Things like the D2, D3, and alpha-1 receptors, which mostly live outside the spines and promote these different detrimental signaling cascades. So these excessive signals quickly shift the network from organized, stable firing to just disorganized, noisy firing.
So the signal is so loud it becomes pure static, and that just impairs all the PFC-dependent functions like working memory and cognitive flexibility.
Correct. And crucially, this impairment is acute and it's rapidly reversible. It happens within seconds to minutes of the stressor starting.
Right.
And the fact that the mechanism is receptor-driven means that the cognitive impairment can actually be prevented pharmacologically. You can just administer antagonists or blockers of these neuromodulators before the stressor hits.
That is powerful evidence. The immediate cognitive crash you feel during an intense moment is a molecular event, not just a psychological distraction.
Yes.
The system is structurally incapable of functioning under that chemical barrage.
It confirms that stress immediately sabotages the PFC's efficiency, using its own regulatory system against it, even before any long-term structural or metabolic changes can begin to take place.
That sets the stage perfectly for the long game. We know how stress breaks the system quickly and reversibly. Now let's transition into how this short-term jam translates into long-term chronic collapse.
And, maybe more importantly, whether that damage, once it's established, is permanent.
Right.
We're moving from the acute physiological jam to the chronic long-term breakdown. For this, we have to look at Section 2, focusing on that critical work by Arnsten and colleagues in 2009.
The study that tried to bridge the gap between animal models and, well, us.
Exactly. They wanted to see if the severe functional impairments they saw in rats who had gone through, 21 days of repeated restraint stress could be cleanly mirrored in healthy human subjects facing natural, real-world chronic stress.
And tell us about their brilliant experimental setup, because finding a natural stressor is tricky.
It is. But they identified a perfect one, a naturalistic, quantifiable, and temporary stressor. They studied 20 healthy young adults who were exposed to 1 month of chronic psychosocial stress while preparing for a major academic examination.
That is a perfect natural experiment. High stress, a common experience, and an endpoint that guarantees the stress will be reduced.
And they could quantify the subjective experience of that stress using the Standard Perceived Stress Scale, the PSS.
Providing a solid anchor. So what was the task they had to do?
Crucially, they designed a task specifically tailored to the PFC functions known to be impaired in the animal models. It's called an attentional set shifting task, and they did it inside an fMRI scanner.
And the genius of the task was its ability to separate different cognitive functions.
Slightly. The subjects viewed these colored moving gratings, and they were cued to respond based on either the color or the motion of the stimuli. This allowed the researchers to isolate 2 key types of cognitive function.
Okay, what's that first one?
The first is attention shifts, which are highly PFC dependent. This required the brain to switch its flexible attention from, say, focusing on motion to suddenly focusing on color.
That's the core executive control function, switching gears, which we know the PFC is responsible for.
Exactly. The second function was response reversals. This involves learning a new rule, but within the same category. So, for example, if you were choosing red, now you must choose blue. And response reversals are of comparable difficulty, but they're mediated by areas independent of the prefrontal cortex.
So if the stress selectively impaired the attention shifts, but not the response reversals, that would be the smoking gun.
It would confirm that the damage was specifically to the PFC, mirroring the rodent findings.
So what did the human behavioral data actually show?
The findings were incredibly sharp and selective. Chronic stress selectively impaired attention shifts. The stressed subjects showed a significantly higher shift cost, which is measured as a slower reaction time and often more errors when they had to switch focus.
And the correlation was strong, wasn't it? The more stress they reported, the worse they performed.
Highly significant. Their individual PSS scores were highly predictive of the severity of this cognitive impairment. The correlation coefficient was about 0.51. In human neuroscience, especially with subjective measures like stress, an R value of 0.51 is a remarkably strong link. It tells us that the feeling of stress directly translates to the measurable inability of the PFC to quickly switch gears.
So we confirm the impairment is real, measurable, and it's proportional to the subjective stress. And crucially, we know it wasn't just general fatigue.
Absolutely not. The subject's general processing speed was unchanged, and the stress did not impair the response reversals task.
That selectivity is the scientific gold standard here.
It is. It means their difficulty wasn't because they were just generally slow or sleepy or distracted. It was a targeted surgical disruption of the specialized executive function managed by the dorsolateral prefrontal cortex, the DLPFC.
Okay, the behavior failed. Now let's see the software failure underneath using the fMRI. What did the functional imaging reveal about the underlying network?
Well, the fMRI confirmed that the attention shift task engaged a robust frontoparietal network, which includes the DLPFC. But the key insight came from analyzing how those regions communicated with each other.
The functional connectivity analysis.
Exactly. We're measuring how tightly correlated the activity is between distant brain regions. Are they firing in sync?
Right.
And in the stress subjects, the DLPFC, the Executive Command Center, showed significant decoupling.
Decoupling. So they're out of sync.
Yes. The DLPFC showed weaker correlations of activity with the very regions it needs to coordinate attention shifts with. This included the right DLPFC, the left premotor cortex, bilateral ventral PFC, and the posterior parietal cortex. These are the regional managers the CEO needs to talk to.
So the stress essentially created gridlock in the brain's executive highway system.
That's a great way to put it. The CEO is issuing commands, but the transmission lines to the rest of the management team are compromised. The signals are corrupted.
And that decreased functional coupling was directly correlated with the attention-shifting impairments.
Directly, physically. The stress wasn't just slowing the DLPFC in isolation. It was isolating the DLPFC, preventing it from integrating information across the necessary structures. The software system designed for complex coordination was failing.
That explains the inefficiency. The brain is trying to solve the problem, but the parts can't talk to each other effectively. But the truly game-changing insight from this 2009 study was the test for reversibility. After a month of high stress, what happened when they retested the subjects after the exam was over?
The findings offered an incredible message of hope. Subjects were retested 1 month later, exam is done, and their self-reported stress levels returned to control levels.
And the cognitive crash.
It disappeared. The attention shift costs returned to control levels, and critically, most of the DLPFC functional coupling measures, that network gridlock, also reversed. It returned to the efficiency seen in the control group.
So the cognitive impairments and the network gridlock were temporary. They were a state-dependent phenomenon, not a permanent injury or some pre-existing trait exposed by the stress.
This is a major finding because it demonstrates the profound plasticity and resilience of the human PFC. It shows that chronic psychosocial stress, even for a month, causes a functional disruption that can be resolved simply by removing the stressor and allowing the system to rest and reorganize.
And this ties back beautifully to the animal models, where the dendritic retraction also reversed after the stressor was removed.
That cross-species validation is essential. The structural damage and the functional failure are temporary effects of the stressor. It strongly suggests the brain is built to recover, if you give it the opportunity.
Okay. We've established the functional failure in humans. Now let's move into Section 3 to understand the physical reality of that compromise, the chronic hardware failure structural attrition.
Right. We hypothesized that the functional gridlock must reflect underlying structural damage. What exactly was happening at the cellular level in those rodent studies?
Walk us through that cellular damage.
In the medial prefrontal cortex, the MPFC, of these chronically stressed rodents, there were profound and specific structural changes to the neurons, specifically the pyramidal cells. First, researchers observed a significant reduction in dendritic arborization.
Dendrites are the tree-like branches that receive incoming signals, right? The neurons receiving antenna.
Exactly. And under chronic stress, the entire dendritic tree literally shrinks or retracts. It pulls back its receivers, which physically reduces the surface area available for synaptic input.
That alone limits communication capacity. But then there's the specific loss of the connection points themselves.
That's the second major finding, a dramatic reduction in spine density. Dendritic spines are these tiny protrusions where the actual synaptic connections from other neurons are received.
And chronic stress caused a catastrophic loss.
Up to 33% of these synaptic connections on pyramidal cells in the MPFC, just gone.
A loss of 1/3 of your communicative hardware is massive. If you lose that many inputs, your neuron's ability to integrate information across the network is just fundamentally compromised.
It is.
This structural loss, this physical thinning of the communication infrastructure, directly explains the functional decoupling, the gridlock we saw in the human fMRI.
The physical attrition is the hardware failure point. If the architecture is compromised, the function cannot be maintained efficiently.
So now let's see how this translates to the whole human brain, beyond just the cellular level. When researchers look at human cohorts facing chronic occupational stress or burnout, what structural signature do they find?
They use a technique called voxel-based morphometry, or VBM, to measure gray matter volume and cortical thickness. And these studies, particularly those on occupational burnout, show a highly consistent structural signature.
Which is?
Generalized prefrontal thinning, atrophy.
So the cortex itself, that thin, folded gray sheet responsible for our highest cognitive processing, is measurably shrinking.
Yes. We see a loss of cortical thickness, often in key executive regions like the ventromedial prefrontal cortex, the VMPFC, and the dorsolateral prefrontal cortex, the DLPFC. This has been consistently reported by researchers like Savick in 2015 and Abe et al. In 2021. It's the physical manifestation of the CEO's office losing its walls.
And it's not just the cortex, our cognitive center. The sources also point to structures involved in motivation and reward.
Correct. Coupled with this prefrontal erosion, we see striatal reduction.
A shrinking motivation center.
Meaning a decreased volume in the caudate and the putamen. The striatum is crucial for habit formation, goal-directed action, effort calculation, and the structural change has been noted in multiple cohorts, including studies by Blix and colleagues in 2013.
That combination, a thinning executive cortex and a shrinking motivation center, is a perfect neurobiological picture of burnout.
Absolutely. The structural evidence is undeniable. But we must understand why the PFC is the primary target. Why is it so uniquely vulnerable to chronic stress?
Right. Let's nail down the mechanism of this structural attrition. You've mentioned the acid rain analogy.
As detailed in Arnsten 2009 and later papers, the pyramidal neurons in the PFC are exquisitely sensitive to the prolonged chemical barrage of stress hormones.
We're talking about glucocorticoids like cortisol.
Exactly, and also excessive noradrenaline. That chronic exposure triggers what is known as excitotoxic pruning. And importantly, it doesn't typically kill the neurons, which is what can happen in other regions.
Okay.
Instead, it triggers this cascade of chemical signaling that is fundamentally neurotoxic, specifically to the delicate dendritic architecture.
So it's not killing the tree, but it's making the tree pull back its branches and shed its leaves.
That's the acid rain analogy brought to life. The stress hormones act like a slow, corrosive chemical process, chemically pruning the dendritic spines and shrinking the apical arbors over time.
And this loss of complexity translates directly to reduced cognitive flexibility and working memory capacity.
Which loops us right back to the behavioral findings in the Liston study. The hardware failure explains the software failure. If you compromise the architecture, you lose the function. The loss isn't random. It's targeted to the complex, high-energy processing structures of the executive brain.
Now, we must address one of the most intriguing and important findings in this area, the repeated observation of a gender divide in hardware failure.
Yes, this is extensively reviewed by Sivik in 2018 and 2025.
The location of this structural damage seems to show sex-specific patterns. Why is this distinction so crucial?
It's critical because it helps explain the diverse ways that chronic stress manifests clinically. While it clearly affects both sexes, the structural signature in exhaustion syndrome often appears lateralized to different functional systems.
So, walk us through the structural signature, often seen as more pronounced in men.
We see that the striatal and PFC atrophy, the measurable shrinkage of the caudate volume and the prefrontal thinning, was often found to be more pronounced or strongest in men suffering from burnout.
And since the striatum is so involved in motivation and effort,
this physical loss maps onto the classic symptom of male-dominant burnout: the collapse of effort, the inability to initiate action, the complete loss of drive.
So the male neurobiological signature under burnout is often centered on the atrophy of the motivational and executive machinery.
That's a reasonable summary of the correlative evidence. But we see a different pattern often dominant in women, and it's centered on the emotional alarm center.
This is the amygdala hypertrophy, the physical expansion.
Yes. Amygdala enlargement was often documented as predominantly, or sometimes strikingly, female-specific in these cohorts with chronic exhaustion syndrome.
So the amygdala, the limbic alarm center for fear and threat assessment, physically expands and sensitizes.
That is a profound physical difference. If the male signature is often the failure of the cognitive engine and motivation, the female signature is often the expansion and hypersensitization of the fear and alarm system.
It suggests two distinct physiological signatures tracking the major axis of burnout symptoms: cognitive and motivational collapse versus emotional hypersensitivity and anxiety.
It provides a crucial layer of nuance, suggesting the primary targets of chronic stress neurotoxicity may differ based on sex, leading to different symptom presentations. And it reinforces the idea that structure dictates function. If the amygdala expands, it's inherently easier to trigger the alarm system.
And if the striatum shrinks, motivation is inherently harder to maintain.
Precisely.
Let's now move into section 4. This is all about what happens when that thinning architecture, that structurally compromised network, is forced to work. This is functional collapse and system performance degradation.
Right, and here we see the brain exhibiting two distinct and paradoxical modes of failure when it's under load.
Let's start with the immediate reaction when the brain is still trying to fight the failure. This is the pushing harder paradox. What does task-based fMRI show?
In high-stress cohorts, so medical residents, for example, doing cognitively demanding tasks, like the Stroop test fMRI studies, showed a highly counterintuitive result.
Which was?
A paradoxical increase in the BOLD signal, especially in the right dorsolateral prefrontal cortex, the DLPFC.
Wait, increased blood flow and increased activation? In someone who feels exhausted, that sounds like they are overperforming, not failing. Why is this a paradox?
Because it signals immense metabolic effort for, often, minimal or no performance gain. We interpret this as the straining computer effect.
Okay.
The executive network is forced to over-recruit metabolic resources. The DLPFC has to push harder, engaging far more strongly than a non-stressed peer would, just to maintain basic baseline performance.
They are using all their currency just to tread water. And what is the observable cost of this high-effort mode?
The cost is extreme mental fatigue and subjective mental drag. The brain is metabolically screaming. And critically, this overactivation often comes with no corresponding improvement in performance or accuracy.
So they're expending 150% effort to achieve 90% results.
Which demonstrates clear neural inefficiency. The compromised hardware requires maximal energy expenditure to perform even routine functions.
That perfectly captures the feeling of chronic mental exhaustion. That overwhelming effort required just to focus on a basic email. The brain is actively trying to compensate for the hardware failure by throwing massive amounts of energy at the problem.
But as the condition progresses, the system seems to realize that this high-effort compensation is unsustainable. And this leads to the second, opposite failure mode, the idling state paradox.
Okay, so in cases of long-term entrenched exhaustion, the people who have been dealing with burnout for months or years, what does their task-based fMRI show?
In these cohorts, demanding working memory tasks, like the two-back test, showed a striking absence or reduction of activation in the DL-PFC and ventral PFC. The BOLD signal just doesn't rise when it's challenged.
It's the functional shutdown. The computer completely shut down the demanding application instead of straining to run it.
That is the interpretation. The system shifts into a chronic, acquired, low-gain idling mode. And this is hypothesized to be a protective measure. The brain has learned the immense, unsustainable metabolic cost of the pushing harder mode.
And it deliberately curbs that cost to prevent a total system collapse.
It's preservation mode, often at the expense of high-level cognition.
So we have two distinct phases of functional collapse. First, the exhausting attempt to compensate, the hyperactivation, and second, the protective shutdown, the hypoactivation. Both are signatures of performance degradation due to compromised neural efficiency.
And this collapse is systemic. It's not just localized to the PFC. The structural and functional damage fundamentally degrades the brain's communication architecture, leading to the concept of burnout as a global connectivity breakdown.
And networkopathy.
A networkopathy, yes.
Let's discuss the brain's internal architecture, specifically this rich club concept. For listeners who haven't heard this term before, what exactly is the rich club?
The rich club is an idea from graph theory applied to connectomics. It describes a small group of highly connected, high-capacity hub regions in the brain.
The major expressways.
Think of them as the major long-distance expressways, or the central internet backbone of the mind. They're crucial for integrating disparate information across the entire brain. And because they're so connected, they're also metabolically very expensive to run and maintain.
And what did longitudinal data, like the studies tracking individuals through periods of high stress, show about the fate of these critical highways?
The data showed a global, consistent loss of strength in these high-capacity, long-range, rich club connections. And this loss tracked the progression of symptoms over time, indicating a systemic degradation of the central command and communication network.
So the stress wasn't just damaging local circuits in the PFC, it was causing the brain's internal structure to reorganize itself into a less efficient system.
Exactly. The system's global topology shifts away from an efficient, centralized hub-and-spoke model toward a more decentralized, less efficient, lattice-like structure. It's like closing all the expressways and forcing all traffic onto slow, local roads.
And that severely impairs the cross-system coordination you need for complex executive tasks. We also have real-time electrophysiological evidence of this gridlock, not just from these slow fMRI signals. The sources mention EEG data.
Yes. A 2025 resting state EEG study provided functional proof of this global coordination failure.
Yeah.
They found that burnout is characterized by significantly decreased functional connectivity in frontal and midline areas, specifically in the alpha-3 subband.
That's 11 to 13 hertz.
Right, and this was during the eyes open condition.
Why is that specific frequency band, 11 to 13 hertz, so important? And why the eyes open condition?
The alpha frequency band, especially the higher range like alpha-3, is associated with the active suppression of irrelevant information. It's vital for selective attention and filtering distractions. So when you open your eyes, your brain is actively challenged to process external input and filter distractions from the environment. A decrease in connectivity in this band means the frontal executive circuits are compromised in their ability to coordinate the necessary filtering.
So this deficiency only emerges when the system is actively engaged and challenged by the non-resting world. It's the physical, electrical signature of the system struggling to manage the basic input volume of reality.
It is.
All of these structural and functional failures, the thinning cortex, the lost spines, the network gridlock point towards one major consequence: the failure of the cognitive brake system on our emotional core.
And that brings us to Section 5, the inhibitory failure and limbic dysregulation.
This is the intersection of cognition and emotion.
It is. In a healthy brain, the PFC, especially the medial PFC in the anterior cingulate cortex, the ACC, acts as the essential cognitive brake on the amygdala.
Which is the limbic alarm center.
The alarm center, yes.
Yeah.
The PFC tells the amygdala, stand down, that's not a real threat.
Yeah.
It sends powerful inhibitory GABAergic signals, GABA being the brain's primary inhibitory neurotransmitter, to regulate and damp down the stress response.
So if the MPFC is structurally thinning, if our brake pad is literally wearing away due to that excitotoxic pruning, that top-down control has to fail.
It does. The chronic stress-induced neurotoxic damage from excessive glutamate and cortisol in the PFC disturbs the structure of the PFC neurons, which impairs this inhibitory action. The result is an alarm system that runs unchecked, a hyperactive and often enlarged amygdala,
leading to those maladaptive, prolonged responses to stressors. Functional imaging studies have confirmed this exact breakdown, haven't they?
Yes. Studies like Golkar et al.
Mm-hmm.
From 2014 used resting state fMRI and found that burnout subjects showed significantly weaker functional connectivity from the amygdala to the MPFC, the DLPFC, and the ACC. The executives can no longer talk effectively or exert control over the emotional alarm center.
Which means the PFC has lost its ability to regulate the emotion center, but how do they link that specific neural disconnection to a measurable behavioral deficit?
They use the acoustic startle response test. This is a reliable physiological index of emotion modulation, where a sudden loud noise causes a physical reflex. They measure the magnitude of the fear-potentiated startle reflex. And they found that stressed subjects were significantly less capable of downregulating their negative emotion compared to controls. The weaker amygdala-ACC connectivity correlated directly with an impaired ability to downregulate negative emotion. The alarm signal couldn't be modulated effectively by the cognitive centers.
The executive brake was not just weak, it was non-functional.
Exactly. The system is locked into a state of hypervigilance because the higher-order circuits have lost the physical and functional ability to issue the all-clear or stand-down command.
And as a final layer of failure, chronic stress doesn't just cause chemical damage. It triggers a measurable physical inflammatory response that accelerates the destruction, the neuroinflammatory cascade.
This brings the systemic failure full circle. Chronic stress creates a measurable neuroinflammatory environment in the brain, characterized by elevated pro-inflammatory cytokines like interleukin-6. And crucially, studies have shown that these inflammatory markers physically coincide with smaller prefrontal volumes.
So inflammation isn't just a side effect, it's part of the mechanism of structural loss.
The evidence suggests that these inflammatory markers actively disrupt synaptic function and promote the loss of dendritic connections in the vulnerable PFC. They accelerate the excitotoxic pruning we discussed earlier. So chronic stress is not merely mentally exhausting. It sets up a physical pathway where inflammation actively erodes the brain's ability to cope.
This entire deep dive has provided a stunning level of detail, tracing the pathology of chronic stress from the molecular tuning knob all the way to global network collapse.
We've confirmed that burnout is associated with a distinct, measurable neural signature. We've traced the mechanistic pathway, starting with acute catecholamine imbalance, progressing through chronic glucocorticoid neurotoxicity, resulting in structural erosion, PFC, and striatal atrophy, coupled with amygdala hypertrophy and functional collapse.
It paints an incredibly detailed and frankly quite alarming picture of the brain under siege. The systematic dismantling of executive control, the loss of architecture, the heightened alarm state, it's profoundly damaging.
It is. But if there is one message that resonates throughout all the source material, it is the constant emphasis on the brain's intense plasticity.
Right.
Despite the dire nature of the damage we've detailed, these structural and functional changes are largely and demonstrably reversible. The hardware can be repaired.
That is the essential message of hope. Let's review the powerful evidence of repair. What exactly did the longitudinal follow-up studies show after stress reduction or rehabilitation was implemented?
They showed objective structural reversal. Longitudinal data, like the follow-ups in the SAVIC studies, showed that after structured stress reduction, the cortical thinning in the PFC and the reduction of caudate volume normalized. They recovered.
That's extraordinary. The brain's architecture can regain thickness.
And what about the hyper-responsive alarm center?
A structural enlargement of the amygdala also reduced after stress abatement. The hyper-responsive alarm center literally shrinks back toward a normal, regulated size, demonstrating that the structural changes underlying emotional hyperarousal are also plastic.
And functionally, the network gridlock can be solved. Absolutely. Targeted interventions, from structured stress reduction to mindfulness training to highly specific biofeedback, all strengthen the system. Even 1 month of dedicated mindfulness training has been shown to strengthen interconnectivity among the default mode, salience, and central executive networks.
It effectively repairs that rich club communication breakdown.
It does. And when we look at advanced techniques like real-time fMRI neurofeedback, it demonstrates durable plasticity in real time. By training subjects how to consciously downregulate activity in hyperactive regions like the amygdala, they are inducing what neuroscientists call Hebbian-like plasticity in the trained circuits.
You are literally strengthening top-down PFC synapses, repairing the executive brake, and restoring executive balance.
The brain is not permanently broken. It is simply waiting for the environment and the metabolic capacity to be restored so it can start rewiring itself.
The depth of this research fully confirms that the profound, overwhelming feeling of mental exhaustion is not a moral failing or simple lack of motivation. It is a protective, measurable physiological adaptation.
It is a response to overwhelming allostatic load. The discovery of that precise, stress-induced low-gain state in the DLPFC that acquired reversible state of reduced neural efficiency where the brain flips into idling mode is the most crucial insight. It suggests the brain is trying to survive.
Which leads us to our final provocative thought for you to consider. If we accept that the brain's low-gain, hypoactive executive state is an acquired defense mechanism designed specifically to prevent total system crash under overwhelming metabolic cost, what does it mean for recovery if we treat that overwhelming feeling of exhaustion, that protective shutdown, not as a symptom to be ignored or pushed through, but as an explicit and measurable signal that the neural architecture is already trying to save itself?
That understanding offers a clear neurobiological target for recovery. Honor the shutdown, support the structural repair, and restore the balance. Recovery is not about willpower. It's about providing the conditions for the brain to execute its inherent mechanism of self-repair.